Well, hello everybody. It's a pleasure to be here. Thank you for having us here. My name is Suyash Prasad. I'm a pediatric intensive care specialist by training. I have expertise and experience in rare orphan disease, uh, pediatrics, neuroscience, and gene therapy. And my current role is the Chief Medical Officer and Head of Research and Development at Taysha Gene Therapies. I'm here to talk to you a little bit about Taysha Gene Therapies, which is a company that's on a mission to find potential treatments for patients with monogenic diseases of the central nervous system. We're using proven approaches to bridge this gap between innovation and treatment. And we have a very experienced team with some of the brightest minds in gene therapy across the globe. So I'll be speaking with you, and my colleague Emily, who heads up patient advocacy and government affairs, is also a speaker. And together we'll be sharing our perspective on Taysha Gene Therapies and our approach to treating Angelman syndrome with a gene therapy approach. Taysha Gene Therapies is on a mission to find potential treatments for patients with monogenic diseases of the central nervous system. So in particular, we're focusing on pediatric neurological diseases, of which Angelman syndrome is one. We're using proven approaches to bridging the gap between innovation and actually providing therapies for children with various neurological diseases, and we have a very experienced team at Taysha with partnering with some of the brightest minds in gene therapy across the globe. I'd like to start with a story about our name, Taysha, as it's central to who we are. When we formed the company, which is really only about a year old, one of the first things we had to settle on was the name. Now, our CEO is an individual named R.A. Sessions, and he is from Dallas, Texas, his family from there. And he has a 9-year-old daughter who happens to be a Texas history buff. And she suggested Taysha because in the Caddo Native American language, which comes from Texas, this actually translates to ally or friend. And this also has an additional meaning of Texas or Tejas. And this resonated with our team, both from a historical perspective from Texas, but also just this whole theme of being allies and friends with various collaborators. We have deep roots and relationships with the biomedical community in Texas. Our world-class partnership with UT Southwestern is an illustration of this, and I'll spend a few more moments talking about that shortly. And we are developing and continue to develop lasting alliances, deep-rooted alliances with the rare disease community, with organizations such as yourselves. And we see this partnership, this allyship, this alliance to be very important and instrumental in how we develop medications and ultimately commercialize them where they can be of benefit to patients all around the world. So this theme of ally and friend really captures the spirit of who we at Taysha are. Now, if you look at how Taysha is built, we have 3 fundamental building blocks for developing our gene therapies quickly and efficiently. The technology we are using has already proven its merits. It is AAV9 gene delivery. So the way we deliver and target a dysfunctional gene has been used previously for a gene therapy for a disease known as spinal muscular atrophy. This was one of the first gene therapies approved in the US. The name of the drug is Zolgensma, and it's treated more than 150 patients with SMA, and the numbers are rising all the time. Now, we chose to work with this technology because we know it's well tolerated, we know it's effective, we know how to manufacture it for commercial use, and we're actually very familiar with all aspects of this AAV9 gene delivery approach. Importantly, UT Southwestern is a gene therapy powerhouse. We are partnered there with a large group both in pediatric neurology and in gene therapy. Stephen Gray heads up the gene therapy unit, and Burj Manassian is the division chief of pediatric neurology. They're some of the brightest minds in the world of rare pediatric neurological disease gene therapy. And in a short amount of time, they've built one of the largest pediatric neurology residency programs in the world and a very large world-class gene therapy facility. So we feel very much so that we partner with the best. Importantly, also, we are building off this technology and research with a key leadership team. that has direct experience of bringing gene therapies through the development process and all the way to a commercial product where patients can ultimately benefit. Indeed, many of my senior colleagues have worked with each other in the past and all have extensive rare disease, pediatric, and gene therapy expertise. The fundamental component of our approach to gene therapy, as I've already mentioned, is an adeno-associated virus serotype 9 or AAV9 capsid. We deliver this intrathecally, and as I say, we have a well understood manufacturing process. Through our partnership with UT Southwestern, we have access to a good manufacturing practice or GMP compliant manufacturing suite. This utilizes what's known as a HEC293 process to produce AAV9. For those that don't know, HEC293 is a mammalian cell line and is the best understood and the most widely used gene therapy manufacturing platform that exists today. And in addition to utilizing UT Southwestern's manufacturing platform, we are actually building out our own facility. So we have plans to do that. We're moving ahead to establish our own commercial scale GMP compliant manufacturing facility under the leadership of my colleague Fred Porter, who happens to be our Chief Technical Officer. He has a lot of expertise in and experience in building out such facilities. And this is, of course, to meet the demand for product candidates receiving marketing approval several years hence. We use intrathecal administration, which involves direct delivery of gene therapies to the cerebrospinal fluid to facilitate optimal distribution and cell transduction within the central nervous system or the CNS. Okay, let me now spend a few moments talking about the Taysha approach to treating Angelman syndrome. First and foremost, we're using a proven and well-recognized approach, the AAV9 vector capsid, which has been shown to work in a number of diseases that are already in clinical trials and indeed in a commercially approved gene therapy product for the brain for disease of spinal muscular atrophy. Now, AAV9 capsid essentially is a shell within which it is possible to package genetic material. DNA, for example, or in the case of Angelman syndrome, a form of RNA known as a short hairpin RNA, shRNA. Now, as you know, in Angelman, the paternal UBE3A gene is silenced in various tissues of the body, in particular in the brain. And the maternal gene has a mutated gene. And so the fact you have a silenced paternal gene and a mutated maternal gene means that there's a lack of UBE3A protein in the brain, which, as you know, has a role in removing a waste product. Our approach to treating Angelman focuses on administering a short hairpin RNA, or shRNA, which upon binding to the target of interest is designed to reactivate the silenced paternal gene, thereby allowing for an increase in production of UBE3A protein, which can then perform its action and generate a therapeutic benefit to the patient. How do we get the shRNA where it needs to go? Well, it's packaged in these AAV9 capsids, and we would inject a number of capsids within a solution into the intrathecal space. Now, what I mean by this is that the brain and the spinal cord, the brain and the spinal cord obviously being the target organ for this drug, they're bathed by cerebrospinal fluid. And the fluid is encapsulated in a space known as the intrathecal space. This space is accessible at the through the spine and is often used to administer various different medications. I myself, in my pediatric practice, administered drug into the intrathecal space. And what happens is that you pop a small needle into the L3, L4 space or the L4, L5 space, part of the lumbar spine. You make sure cerebrospinal fluid is coming out of that particular needle, which means you're in the right spot. And then you attach a syringe driver with some tubing and you inject maybe 10 mils of the drug. You then take the needle out, you tilt the patient back so they're recumbent with a head down position, about 30 degrees down. And the cerebrospinal fluid therefore bathes the brain with this drug, and you get drug entering the brain, entering the cells of the brain, and exerting its therapeutic benefit. Importantly, we'd like your help in understanding the disease of Angelman syndrome as we get closer to a clinical trial, especially what you feel are the most difficult features of the disease and what you hope to see in improved motion from the treatment. So on this slide, I know you know the clinical features of Angelman very well, but the kinds of things we can study in the clinical trial are seizures. How often are the seizures? How long do they last? What's the severity? What medications do you use? We can look at developmental progression of the child, i.e. how the child is developing cognitively, or how the child is acquiring their gross motor milestones, the ability to stand, the ability to walk, the ability to jump on one leg, or indeed fine motor milestones, the ability to pick up a small pea off the ground or pass an object from hand to hand. And of course, speech and language development is critically important for us to observe and monitor within a clinical trial. We can also look at features such as sleep. There's new technology in form of wearables that help us look at sleep architecture in more detail. And of course, we're looking at safety and tolerability within the context of a clinical trial with the gene therapy medication. But importantly, as we get closer to a clinical trial, we want to be talking with you all in the patient community because we really want to understand which of these clinical features are most important to you as parents of children with Angelman and indeed to your children themselves. This helps us in our conversations with regulators, and it helps us prioritize the endpoints in terms of how important certain clinical features are and what clinical features are we hoping to see an improvement in. I want to remind you that we are actually in the early stages of the development process, and I'm very pleased to be here, but we do have some way to go. The drug development process itself can be quite long, from discovery and early preclinical work to clinical trials and potential regulatory marketing approval. That can take a number of years. With regard to our preclinical work, the kinds of things we do there are we actually define and select a construct, i.e. what is the actual molecular structure of the gene therapy vector, and then we start dosing in the animal context. So we look at different doses to see how effective the gene therapy drug might be and also how safe it might be in order to build out a therapeutic window. What's the lowest dose to get an effect and what's the highest dose from a safety perspective, and then we have a range within which to play for our clinical trial. At which point we'll start the clinical trial, and as I say, we'll be talking with you about that as time progresses. Once the clinical trial is underway and we are starting to accumulate data, when we have enough data from the clinical trial demonstrating the safety and the efficacy of the drug, we will then talk with the FDA and sometimes the European agencies at the same time and see exactly how much information they need for us to file for marketing approval. And hopefully, we'll run the clinical trial. Hopefully, it won't be too long, too extensive, and we'll see a nice improvement in the children with Angelman syndrome, and then we'll be able to file for an approval with the FDA. Throughout this development process, we see the patient community as our partners, as our collaborators, and our teachers. We look forward to learning from you, as well as providing updates to you as we have more information to share about the program. I'd like to thank my colleagues at Taysha for helping out with this presentation. Obviously, I'd have loved to have been there with you in person, but we're doing things remotely for the time being. I also want to thank my collaborators and partners at UT Southwestern, so Steve Gray in particular, Virgin Manassian in particular, and Ryan Butler, who is leading the Angelman effort, and he's an assistant professor at UTSW. Thank you all for listening. Hopefully you soon. And all that remains for me to do is to introduce my colleague Emily McGinnis, who heads up patient advocacy and government affairs, and she can tell you a little bit about the Taysha approach to patient advocacy and how we anticipate working with you over the longer term. Thank you very much.
Hi, my name is Emily McGinnis, and I'm the Chief Patient Officer at Taysha Gene Therapies. While I would love to meet you all in person today, I hope that we'll be able to meet next year at the conference. I previously worked for the company that brought gene therapy to patients for spinal muscular atrophy. Having gone through that experience and witnessed firsthand the benefit that gene therapy brought to this community, I'm really excited to have an opportunity to help other families, such as the Angelman community, benefit from gene therapy. At Taysha, the patient is central to our work, and we do this through patient guiding principles. Collaboration is really key. Taysha was founded on the basis of collaboration, and we are focused on coordination with patients, caregivers, advocacy partners, clinicians, and government stakeholders. Transparency is also another key element where we want to be true partners to our stakeholders and provide proactive communication that is timely and informative. We also approach our work with curiosity. By investing a deep-rooted understanding of the entire patient community and disease pathway, we're able to develop the best solutions for the community. Empathy is also extremely important. We know that the patients and families matter most, and we're fully committed to providing compassion, empathy, and care beyond the product itself. It's actually something we look for in candidates that we bring onto the company. Lastly is respect. Taysha honors the privacy of patients and families by strictly adhering to policies that protect their personal and medical information. You may have heard this before early in the presentation, but I want to reiterate, there are 3 key groups that need to work together to bring therapies to patients with rare diseases. First and foremost, advocacy partners such as FAST, research and industry. Strong advocacy in patient communities like FAST can help advocate for patients, raise awareness of the disease, and make sure they receive the attention they deserve. And like FAST, actually do important research that we can benefit from as well. Ties to academic and clinical research centers ensures discoveries and science moves forward. And lastly, industry provides support and resources and expertise to navigate the regulatory and commercial pathway. For me, this partnership has never been more apparent as we're working hand in hand with UT Southwestern and the patient groups every step of the way. We are truly embracing an integrated approach, one that will help get new medicines in the hands of patients in the fastest and most effective way possible. This partnership with the Foundation for Angelman Syndrome Therapeutics, or FAST, and the entire patient community is going to be critical to Taysha's Angelman Syndrome program. I'd like to start with some ways that you can help work with us in the future. 1, you can help Taysha understand your community, help us understand the unmet needs of your kids and your families. You can also help us understand the areas of educational need for families and clinicians about our gene therapy program. With this information, we can develop clinical trials based on patient and families' insights. And these interactions can help bring awareness to our clinical trials and make enrollment more efficient. Together, we can advance new therapies for Angelman syndrome. At Taysha Gene Therapies, we believe the patient always comes first. The patient, their family, caregivers, and the communities we serve are the foundation for everything we do, from preclinical drug development through commercialization of new treatments. We remain steadfast in our commitment to understanding the unique challenges faced by the communities we serve, keeping those stakeholders' best interests at heart as we seek to deliver impactful and life-changing therapies for Angelman syndrome. To learn more about Taysha Gene Therapies, please feel free to visit our website at www.tayshagtx.com. I really want to thank you all for taking the time to meet the team and a little bit about our Angelman program, and we look forward to continuing to work with you. Thanks.